Hello, good morning. My name is Dr. Prithvi Mruthyunjaya. I'm the Director of Ocular Oncology here at Stanford University Medical Center at the Byers Eye Institute in Palo Alto, California. Thank you very much for this kind opportunity to be part of this really global event with many friends and colleagues who are participating, and also our patients who have taken the time and the interest to be a part of this group. My talk today will be based on the work of the Collaborative Ocular Oncology Group and discussing data safety and monitoring in clinical trials. The Eyes on a Cure Global Symposium is really a unique opportunity to bring patients together to help understand the various complexities of care and how we as clinicians and scientists are trying to move ahead our field. Here are my disclosures. The goals of the Collaborative Ocular Oncology Group is to provide evidence-based, investigator-initiated, precision companion diagnostic and prognostic tools to ultimately develop precision therapeutics to attack the problem directly and most efficiently in our patients with ocular melanoma. The investigators of this group span the United States and Canada and represent the preeminent ocular oncology centers, both in academics or at universities, but also in private practice. The history of the Collaborative Ocular Oncology Group begins in the lab of Dr. William Harbour, who is now at the University of Miami. The study originally had a prospective 12-center validation study of the gene expression profile platform. 459 patients with uveal melanoma were included, and this study demonstrated the superiority of the gene expression profile classification over tests like monosomy 3 and clinical features for prediction of metastatic risk. This brought into our vocabulary terms like class 1 and class 2 as a way to describe low and high risk melanomas and the risk for metastasis. Over time, the work from Dr. Harbour's lab has really blossomed, and now we learn other terms in our lexicon, including terms like class 1A, class 1B, and also the presence or absence of PRAME. These are all thought to be biomarker signatures within the tumor that's helping we as clinicians understand risk of a melanoma progressing to something more serious, such as melanoma spreading to other parts of the body. The third funding iteration of the COOG study brings us to our current study that we are doing now called the COOG2. This is a prospective study to evaluate additional prognostic biomarkers, specifically the PRAME antigen expression as an independent predictor of metastasis in uveal melanoma, and also to look at what we call next generation sequencing. So the most advanced types of genetic and molecular identification of risk factors that will help us to determine whether there are ways to determine risk even without doing a biopsy of the tumor in the eye, maybe just of the fluid of the eye or even blood. The way this study works is that we have clinical centers that enter patients. Many of you may actually be in this trial, and we thank you for your participation in this important study. Samples are sent to Castle Biosciences in Texas, who perform the gene expression profile test and give us results, but also help run a validated next generation sequencing panel. And that information is independently put into a database that's secure, and only then is it analyzed. And this keeps the clinicians separate from the laboratory group, from the investigators, so there is no bias or overlap in any of this. The current COOG leadership has been developed and really demonstrates the importance of collaborative working together and bringing in ideas from different parts of the country and different perspectives. Dr. William Harbour is the chair of the current steering committee, And many prominent ocular oncologists, many who have treated patients who are on this seminar today, include Dr. Thomas Auberg from Michigan, Dr. Zelia Correa, formerly at Johns Hopkins, but now at University of Miami, Dr. Brian Marr from New York City at Columbia, Dr. Amy Scheffler from Houston, myself at Stanford, and Dr. Jose Lutsky, who is a medical oncologist, not an ocular oncologist, who brings his expertise to us from the University of Miami as well. The medical oncology subcommittee is made up of three prominent medical oncologists, Dr. Richard Carvajal from Columbia, Dr. Lutsky again from Miami, and Dr. Takami Sato from Thomas Jefferson. I'm also honored to serve as the chair of the data committee in this group. I'll give you a quick update on what we've learned so far from the COOG2 study. As of October, over 1,600 patients were enrolled. So thank you to each and every one of the patients that were entered into this study. It is now closed for recruitment. And we know that about 51% of the patients were female. The median age was 62 years, and right eyes made up about 53% of all the eyes studied. Importantly, we have gene expression profile data on over 1,550 patients, which makes us one of the largest prospective studies ever conducted in ocular melanoma. So we are very pleased by the response that we've had and the participation. We found that two-thirds of the patients were class 1, or the lowest genetic risk for metastasis, and a third were the higher risk class 2. And this is consistent based on a lot of other papers that have been published. So this validates that information. Class 1A was about 64% of the class 1 patients, and class 1B was 36% of them. PRAME status, which is the new biomarker that's being validated, was found to be present or positive in 30% of this cohort. So 70% didn't have PRAME. You can see now we have incredibly large populations of patients that we can use to compare to one another. The future goals is an integrated precision medicine instrument that we can develop from just taking a biopsy of the tumor, or maybe the blood, or maybe some of the fluid from around the eye. This information from this study will tell us a lot about these biomarkers, will help us to understand how patients should get tested for metastatic disease, how frequently they should have surveillance. We will also use this science to determine whether there are other therapies that we can give to patients who are at high risk, to see if this can eliminate or alter that risk, and we call that adjuvant therapy. And finally, do therapies that we give to our patients, can they be so customized that specific mutations within a patient might determine what therapy they get? Personalized medicine, that would be the goal in the future. What are the purposes of a clinical trial, though? One is to advance knowledge, but also to do it in the context of safety, but also finding effectiveness in the purpose of the study. The trials are put together based on available science, but they have to be executed. And let's just talk about that, because many of you have asked about what are clinical trials, how do you get involved? And then there's always this unknown about clinical trials. In this day and age, we hear a lot about exciting clinical trials in the world of the COVID-19 virus. There are information coming out all the time about clinical trials and safety and whether it's effective. So we all are becoming experts, whether it's through the media or the newspapers, on clinical trials. There are several important participants in a clinical trial: the investigator, the person that puts together the idea of the trial, the sponsors, the companies or organizations that fund and direct the technologies in the trial, the institutional review board, and the data safety monitoring committee. But the most important is the patient, which is you in this trial, and everything hinges on the patient. The investigator is a researcher or group of researchers who organize and conduct the study. They are part of a team. This can be clinicians, oncologists, ophthalmologists, research scientists, statisticians, clinical coordinators, ethicists, pharmacists. It's a huge team. They are responsible for the ideas and ultimately the execution properly of the study. The sponsor, this is the entity that basically funds or organizes the study. This is most likely in drug trials or say COVID-19 vaccine trials, a pharmaceutical company, but it could also be a foundation. It could be a government agency like the National Institutes of Health, which helped to fund the COOG2 study. They may be providing money for expenses, experimental drugs, infrastructure, a whole bunch of things, but they usually are providing the ability for the investigators to execute the study. Now, they could have a vested interest in the outcome. So it's important that the investigators and the sponsors and the patient have a healthy difference or separation so that the desire of the sponsor for their treatment to be effective and be available for patients broadly, but also for purchase, does not cloud the integrity of the study itself and the outcomes. To protect patients, we use several avenues. One is called the Institutional Review Board. This is institution specific. I have my own, MD Anderson has their own, a private office may have their own. But basically, it is an internal review of the trial before it begins. A protocol is written, and the IRB looks at the design, the implementation strategy, the risks and the benefits to the patients, and ask really tough questions whether this trial is worth conducting at an institution. Informed consent is the form that you sign after everything has been explained to you, and that has to be very clearly delineated for the patients. And most important, local level safety to make sure that things are safe as it sounds and as it looks within that institution. The Data Safety Monitoring Board, or DSMB, is a group of outside experts, usually an ethicist, a statistician, medical experts in the field, and maybe even a patient advocate. So a DSMB in uveal melanoma may have a uveal melanoma patient who's not a physician, who's there on the board to review aspects that may be very personal to a patient going through this treatment. The key is they are not part of the study investigator team. They are not entering patients into the study. They're not vested in it. They're not part of the sponsorship of the study. So clearly independent. They look at safety and efficacy across the trial, not just at my institution, but all the institutions. And they kind of have an ability to look at things from up top. They try to make sure that things are safe. And if something looks not safe at one institution, but may apply to others, they can actually stop the trial or put it on pause or halt it till things are figured out. And finally, they look at the efficacy independent of the investigator. So if they see a positive signal, they can tell the committee that we should alter the course, we should move faster, move slower, or stop the study. So they have an incredible ability to influence the readout of the study along with the investigator. So at the end of the day, the patient is the center, and the investigator or your doctor is who you interact with most directly, but the investigator is working double, triple overtime, working with the IRB and the Data Safety Monitoring Committee, but also the sponsor as well. And there may be interactions between the sponsor and the patient and the investigator and the patient, but the key is to keep these groups engaged, working together, but also healthy separation so there isn't overlap that may cloud the judgment of the outcome. In the COOG2 study, since we are not part of a investigational therapy at this time, my committee is looking at the accuracy of the data that's coming in. We do quality checks on the genetic and clinical information that's reported, and we act as an independent verification of the data that is being presented. And finally, we help the investigators ensure that the conclusions are being supported by the data. And all this will help us guide further investigation to promote and progress the treatment options available for our patients. I wanted to thank you for this opportunity to present today, and I look forward to all the other great talks during this conference. Thank you.